In the absence of CAMP-CRP complex, the lag promoter is quite weak because its -10 box differs significantly from the consensus sequence. A mutant lag promoter with a -10 box that has the consensus sequence doesn't require CAMP-CRP complex for transcription activation. It therefore seems reasonable to propose that interactions between CAMP-CRP complex and RNA polymerase holoenzyme increase the holoenzyme's affinity for the lag promoter. Thomas Stitz and co-workers determined the crystal structure of CAMP-CRP complex bound to DNA. CRP consists of two identical polypeptide chains of 209 amino acid रेजिड्यूज. तो इसमें दो पॉलीपेप्टाइड चेन है, हर एक पॉलीपेप्टाइड चेन में 209 अमाइनो एसिड्स जो हैं वो प्रेजेंट हैं. ईच चेन कंसिस्ट्स ऑफ an N-terminal domain and a C-terminal domain. The N-terminal domain contains a pocket for binding CAMP, while the C-terminal domain contains a motif that binds to DNA. तो C-terminal domain के साथ DNA bind करता है, जबकि N-terminal domain के साथ जो है वो CAMP bind करता है. Here you can see this is the structure of CAMP-CRP complex along with the DNA. तो ये इसके ऊपर जो है ये DNA लगा हुआ है. इसमें ये यहाँ पे जो है it is low affinity CAMP site, और इसी तरह ये जो है this is high affinity CAMP site. In the absence of CAMP, CRP DNA interactions are non-specific and weak. यानी कि अगर CAMP नहीं है तो DNA और CRP के दरमियान में जो इंटरेक्शन हो रहा है वो नॉन स्पेसिफिक होगा और वीक होगा. The CAMP-CRP complex, however, binds very tightly to a specific DNA sequence designated as the activator site. In the lac operon, the center of AS is 61.5 base pairs upstream from the transcription start site. Promoters like the lac promoter that have AS at positions -61.5 are designated as class 1 CAMP dependent promoters. Binding of CAMP-CRP to the consensus sequence is so tight that an operon containing this sequence would be permanently switched on. It is therefore not surprising that actual CAMP-CRP activator sites differ from the consensus sequence. Activator sites in different operons compete for the CAMP-CRP complex, with the activator preferentially binding to the sequences that most closely resemble the consensus sequence. CAMP-CRP bound to DNA migrates in an anomalous fashion when subjected to non-denaturing gel electrophoresis. The most likely explanation of this is that the protein causes the DNA to bend when it binds to it. तो जब ये CRP कॉम्प्लेक्स जो है बाइंड करता है DNA के साथ, तो DNA को बेंड कर देता है. X-ray crystallography studies confirmed this interpretation showing that CAMP-CRP complex sharply bends DNA by an angle of between 80 to 90. In addition to specific contacts with activator site, the CAMP-CRP complex also makes specific contacts with RNA polymerase holoenzyme.